IDs of all the students. Otherwise, I would have sent a calendar invite so I don't have to, manually admit each one of you inside. So I would request you that throughout the lecture time, whenever it is possible for you, please just fill in this form which just asks your complete name and email address. Please, if you have your Somaya ID, just put that in so that I can send you a calendar invite for all the lectures and I don't have to manually admit each one of you inside. So, that will save a lot of our time. So please fill in the form. I request you to do that. You can do it during the lecture tenure and fill it up so that from the next session we don't have this issue. Okay, so very good afternoon to all of you. My name is Sachin Rajgopalan, and I am currently working as an assistant professor in the Department of Microbiology at Ramnarayan Roya Autonomous College. So I will very briefly introduce myself to you guys, though I feel that most of you will also be, most of the batch is over here for the elective paper because I'll be dealing with both your core as well as elective paper. So I have completed my bachelor's and master's in microbiology from SIES College, Sion. And after that, I started working as a research intern at a company called as Advanced Enzyme Technology Private Limited, which is in Thane. So I was there in the application development lab. And post my tenure there, I cleared my NET and SET examination in life sciences. And that's how I came into the field of teaching and joined the Department of Microbiology at Ruya College. So it's been 5 years that I am at Ruya, and I teach undergraduate and postgraduate students in the subject of microbiology at Ruya. And I also deal with a vocational course, which is bachelor's in vocational sciences in pharma analytical sciences. So these are the areas in which I have been involved at Ruya College. And other than that, I have my PhD going on simultaneously in the area of biofilms and multi-drug resistance. And other than that, I have guided around 30 students now in master's dissertations. And my area of research basically lies in quorum sensing, biofilm studies, enzymes, industrially significant enzymes basically, and very recently have started exploring the area of multiple drug resistance since I've started doing my PhD there. I have been trained in molecular biology techniques from Indiana University of Pennsylvania, USA, and have been working on a project in collaboration with them, and also have represented the country in an international genetically engineered machines competition, wherein Ruya had a team in 2018, and we had genetically engineered E. coli to degrade plant stains. So we had one gold medal in that event for India amongst 385 global universities, and also a Best Human Integrated Practice award. So yes, it's been a journey which has had mixture of academics as well as research, and I'm very glad to be part of Somaya's initiative here of having us for the course of undergraduate as well as postgraduate studies as guest lecturers. So I will be dealing with 2 units from your elective course on animal biotechnology, and I'll be also dealing with 1 course from your core paper. But I'll not discuss about the core paper today. We will have a discussion on about that topic on Saturday when we actually meet for the core paper. And today we will restrict our discussion only to the units that we will be dealing with in this elective paper. So obviously, today being the first day, I don't want you to be burdened with a lot of concepts. That's not the aim. But yes, I want you to know what are we going to learn and why are we going to learn. Until and unless we have this understanding of how is this stuff going to be useful for me, whatever I'm learning right now at this point of time, where am I going to use it? How is it going to make the human life better in any way? Until and unless that is clear in our mind, it doesn't invoke the interest in the concepts that we are going to be dealing with. So that's the reason why what I am going to do is I will take you through the different concepts that we will be learning in these 2 units. I will also tell you what my methodology is going to be in the teaching learning process that we have. And I will also tell you a little about how I'm going to share the study materials with you guys, because of the cross domain, as I have my email ID on Ruya College domain and you guys have an email ID on the Somaya domain. There is a problem creating the classroom. So that's the reason why what the plan is that I will be generating a wiki link. What a wiki link is, I will let you know by the end of today's session. And I will also share it with you guys so that you have the details with you and you don't have to struggle finding things at a later point of time. Okay, so I'm very excited to have a really productive interactive sessions with you guys, because please remember, these are not going to be one-way sessions. These are going to be more of interactive sessions where I would want you guys to speak. Okay, please do not feel shy, do not be afraid. We guys are here to learn, not just you guys, even me. I am here to learn. We all are from the learners community. Learning never stops in life. Okay, so please do not feel shy to ask questions. Whatever comes to your mind, feel free to speak that out. Feel free to quote your questions, and I'll try my level best to answer those. I don't promise you that I will have answers to all the questions that you may ask me, but I can ensure you that even if I don't know something, I will get back to you once I read about it, because no one on this globe is perfect, right? So, with that, let me tell you that I always believe in this fact that no question is a chindi question, okay? The questions that you ask, the questions that you raise, they may be the simplest of the ones, but always in the history, if you see in the history of science, you will see that the simplest question has given rise to the biggest discoveries that we have today. So don't stop yourselves from asking questions. Don't feel afraid. Don't feel shy. Come up with your questions and we can have discussions on it. All right? Okay, great. So with this, let us start discussing about the 2 units that I will be dealing with. The very first unit that you will see that I am going to be dealing with is on transgenic animals. Okay, and this particular unit is quite interesting for the kind of concepts that you will be learning in this particular section. Now, first and foremost, can someone tell me here as to what do you mean by transgenic animals? Can you tell me that? What are transgenic animals? Come on, it's fine if you go wrong, not a problem at all.
So
Sir,
genetically modified.
Okay, genetically modified. Yes, Namrata, you were adding something there.
sir, I was saying that the animals in which a transgene is added, those are transgenic animals.
Okay, so can you tell me what are transgenes? A very good way of describing what are transgenic animals, but what are transgenes? What do you
Sir, the genes which we are desiring, like that we want a particular gene in a like other animal. That's why those genes which are transferred from one animal to another are called transgenes.
Excellent. So, with the advent of recombinant DNA technology, a lot of things changed, right? And let me take you through that journey. I hope you guys would be okay with me giving you some examples so that you can relate to the concept. Is that fine with all of you? I will take some time, deviate through the topic, and then come back. Is that fine with all of you?
Yes,
Yes,
sir.
sir.
Great. Okay.
Yes, sir.
So when we talk about recombinant DNA technology, we know that this technology has brought about a lot of change, right? Now let me give you a simple example that how this technology can actually bring about huge changes in terms of commercialization. Okay. So first and foremost here, you all know about vanilla ice cream, right? We all enjoy eating vanilla ice cream. But let me tell you that the vanilla ice cream, basically the taste, the flavor of vanilla comes from a component called as vanillin. This is that component which gives you that flavor of vanilla. Okay. Now, vanillin is extracted from a plant. It's called as vanilla pods. And You know that all plants cannot grow everywhere. They have specific climatic requirements, they have specific geographical requirements. And vanilla pods are something which grow luxuriantly in Australia. And if you have those vanilla pods, if you're going to extract vanilla out of it, or rather vanillin out of it, and you're going to use it for the process of making the vanilla ice creams and giving it that flavor, it's going to create a hole in your pockets because vanillin is damn expensive. Okay? So what people did, they switched to the concept of essence. We use this term very commonly, right? That you make something which has a flavor of pineapple, it is pineapple essence. Similarly, you have some biscuits, cream biscuits made up of mango, it has the mangiferin essence in it. Essence is basically nothing but chemical equivalents of that particular flavoring agent, which is created in the lab, and you are making use of it, to give flavor. But it's not originally the same component extracted from the natural fruit. No. Okay? So whatever vanilla products we see around us basically contain the essence, which is nothing but chemically manufactured flavoring agent in the lab. Because purchasing vanillin is going to be pretty expensive. It's as expensive as paying 35,000 bucks for 1 kg of the vanillin extract. Now, if Amul starts making use of this vanillin extract, vanilla ice cream would become a delicacy wherein we have it once in Diwali. That's not the case, right? That is not the case because we are making use of the vanillin essence and not the original vanillin extract. But now what if I tell you that there are institutions which have fished out the genes from the vanilla pods which are responsible for synthesizing vanillin? Insert that gene into E. coli. What is it going to do? In 24 hours of its growth cycle in the fermenter, it is going to give you tons of vanillin extract. And that is what can be used as the original vanillin essence because it's the same gene which is seen in the vanilla pods. And the best part is that 1 kg of such a vanillin extract which has been produced by our DNA technology is going to cost you between 35 to 40 rupees. Where is 35,000 rupees and where is 35 to 40 rupees? So that's the basic difference that we bring about in terms of manufacturing at a low cost, maintaining the quality using our DNA technology. Now, there are several products which I may not be able to generate in microorganisms for several reasons. That could also be because those products are such that it kills the microorganism itself which is producing it. Then, if the cells are going to die, my production will stop there. And then obviously the yield that I get from such a process is going to be very low. So under such circumstances, I may have to express this product in a different system. And those systems could be animal models. And let me tell you that you may think that, is it really done? Indeed. Let me tell you that plants and animals, since a very long period of time, have been treated as biofactories. It's not just microorganisms. We are under that notion that probably it's only the microorganisms which can be utilized to, produce the product that you aim to commercialize. No. There are plants, there are animals which are being manipulated so that you are able to get a desired product. No doubt that doing this with microorganisms would have been simple, easy to handle, right? Because microorganisms, though we say are fussy, at some point of time with our DNA technology, we know that we can manage it. But if you're talking about a plant or animal, there are so many factors that come into the picture. Number one, their growth cycle. Number two, how am I going to introduce this gene? Will it be delivered at the exact site? Will it give me a yield as good as what I get from microorganisms? There are so many questions that would come up when we talk about using plants or animals as our biofactories. So that is exactly what we are going to focus on. That is exactly what we are here to study. But unfortunately, whenever we talk about transgenic animals, I have seen people looking at them only as commercial models. That हाँ हमने ये बनाया है, इससे हमको कुछ प्रोडक्ट मिलने वाला है, उससे बेचकर हमको पैसे मिलने वाले हैं. बट ये सच नहीं है. ये सच नहीं है क्योंकि हम ट्रांसजेनिक एनिमल्स डेवलप इसलिए भी करते हैं बिकॉज हमको एक डिजीज मॉडल चाहिए, एक जेनेटिक डिसऑर्डर मॉडल चाहिए. Otherwise, how are you going to know that what happens in a specific physical condition inside a human patient? So if I want to do that kind of a testing, I need an animal model suffering from that kind of a disease or that kind of a disorder. So for that also, transgenic animal models are designed. Now, best example I could give you is when we talk about people suffering from neurological conditions. You talk about Parkinson's, or you talk about Alzheimer's. Have you heard about these two neurological conditions in the past?
Yes, sir.
Yes or no?
Yes, sir.
Yes.
Yes, sir.
Very good. So do you know what Parkinson's is? क्या होता है? मतलब it's okay if you don't tell me which gene is involved. That's not what we are discussing about right now, at least. But what happens in the condition?
Chaperone dysfunction,
And what's the consequence?
abnormal.
I couldn't hear you clearly, Himali. Can you repeat?
That nerve function affects due to that.
Okay, not
Neuro-
exactly, but I can tell you the consequence here in brief is that, yes, Devendra, do you want to say something? Okay,
So tremor happens.
very closely related to what Himali has mentioned. The basic effect that you see in case of Parkinson's is that mobility is affected to a great extent because of neurological damage and muscular motor functioning damage. Now, in them, in the beginning of Parkinson's, when Parkinson's just sets in, what do you see? You see that they're not able to move their hands properly. The flexibility that you have in your arms, the flexibility that you have in your fingers is lost. Okay? You will see that even when they walk, they are not able to take longer steps. No, they feel restricted in their movement. They are able to feel that something is holding them back from moving. You know, that's how the Parkinson's sets in, wherein they will feel that they are not able to move. This then is accompanied by instability of arms and legs, wherein they will feel that they are not getting balance. They feel tremor, as someone has written in the chat box, as shivering. Okay, and gradually they are not able to exhibit mobility. They are not able to move. So what happens to them? They become localized. Okay. Now gradually, as their hands and legs movement is stopped, you will also see that other movements of the body starts becoming sluggish gradually, like moving the neck. So they will not be able to very easily move their neck as we are able to do it in either of the directions, or up and down as well. Gradually, you will also see that their tongue movements also starts getting affected, because again, a muscular coordination there, right? Their tongue movement starts getting affected. So the speech becomes, slurry, like, wherein it's not clear what they are trying to say. Then you will see that they are not able to move their eyes. Okay, and here I am talking about the movement of the eyeballs. Seeing in either directions, up or down, becomes difficult. Closing, opening eyelids becomes difficult for some. And gradually, what gets affected is the heart. So you will see that Parkinson's sets in the late 40s. For some, even after 35, it sets in. And it doesn't lead to death of the individual immediately, So that's the reason why it becomes more frustrating for them. Because you see that gradually your condition is progressing towards the worst, but there is not much that you can do because you don't have medications to cure this condition. There is no cure as of now. What you can do is you can slow down the progression to a great extent, but you can't stop the progression. The person is going to progress towards that stage. Though today we say that a lot of gene therapeutic methods are being tried out, but none of these have been 100% successful, giving us a very high success rate. There are new therapeutic modules that are coming out, but they are not in practice as of now because some of the other problems are associated to it. And you know that any kind of new therapeutic strategy to come into the market takes a huge amount of time because it needs to be tested, it needs to be proven as safe, and then you get FDA approval, then it comes into the market. It takes a huge amount of time. And along with time, there needs to be a lot of monetary investment. If you don't have investors, all this is going to take even more longer period of time. So if I want to study something like Parkinson's, let us say that whether he tells me that her lab has come up with a new chemical agent which she feels has the ability to slow down the progression of Parkinson's to a great extent. Now to check this, I cannot try this out directly on humans. It's not allowed because I don't know if there are any kind of safety related issues due to toxicity of that agent, because I cannot say to a human that I'm going to try this on you, but you know what, if you eat this drug, you're going to die. No one is going to come for the trial, right? And it's not ethical. There are so many ethical issues associated there. So then what has to be done? I have to test it first on cell lines. I have to test it on animal models. But to test it on animal models, I need to have a mice which is suffering from Parkinson's. So then I have to generate such a mice model. So that once I have a mice model which is suffering from Parkinson's, then I can try using the agents which probably Vedai's lab has developed to see if that agent works in slowing down the progression of Parkinson's or not. So even for generating such disease models, we can make use of this technology. Parkinson's may be just one example. You can see in your syllabi, they are speaking about an even more interesting neurological condition, which is Alzheimer's. So if I tell you that I can create a disease model for Alzheimer's using this technology to test if, the newly available agents, which the pharmaceutical companies are claiming can be really very good for Alzheimer's patient. If I want to test that, I need to have an Alzheimer's model. So I can develop that using this technology. Right? So that's the reason why the labs which specialize in a specific condition, in a specific health condition, they will normally have some kind of disease models, not necessarily mice models. Mice is the most common disease model that you will see, but it is also a possibility that there might be other animal models also. Okay. Other than that, what are we going to use the transgenic animals for? So no doubt we are going to use them as disease models. We are also going to use them for expressing very important pharmaceutical agents, which have a lot of significance therapeutically, and I'm not able to synthesize them in other living models. I will try to synthesize them in transgenic animals. Transgenic animals have also been designed to develop organs which can be used for organ transplantation as well. Transgenic models have been created to develop livestock, cattle, domesticated animals, which can be resistant to specific diseases. You know that several countries, these are considered to be agricultural countries, or they are considered to be dairy economies because their complete economy runs on dairy products. So if they have cattle and they have to have good yield of milk from them, in those circumstances, you have to ensure that the animals remain healthy. If they are infected by some or the other infectious agents time and again, and if that infectious agents quickly spread amongst the cattle, the yield is going to be affected very badly and the economy is going to drop. I would not want that. So can I have this technology work in a way such that it helps me develop a livestock which is resistant to most of these infections? Also, these days, these technologies are being used to increase the yield, not just in animals, but also in plants to increase the crop yield. Also, this has been utilized to increase the milk yield in milking animals. Also, to have better traits. You know that whenever there is crossing that is taking place between two animals, and you want the best possible qualities or characteristics in the young one, you would want the parents to be the fittest. But now with the advent of genetics and sequencing technology and genetic engineering techniques, it has been possible for us to identify that which genes are responsible for a specific set of characteristics and how I can introduce those genes into the animal in whom I desire these characteristics, so that I don't have to wait for the proper breeding to take place, where I ensure that the parents are healthy, they have the best possible characteristics, then I cross them. Not needed. So I can do that using the transgenic techniques that are available today. So is it just in cattle that it is done? No. We are using this technology for developing transgenic poultry as well. Maintaining a proper hygienic poultry farm is a very difficult task, because they have this tendency to fight with each other. Okay, when they fight with each other, they end up creating scratches on each other's body. Very prone to getting infections if you are not careful about hygiene. And once one poultry, in a poultry farm, one animal is affected, barbad hai pura poultry farm. In no time, you will see that all others are affected. That's the reason why the poultry farmers, they're so careful. They constantly keep a check that even if one of them is showing any disease symptom, pick them up, separate them, quarantine them, because in no time the others will be infected. So you have to be very careful. But if I develop transgenic poultry, which has the ability to resist most of the conditions, most of the infections, then the work is going to be very simple. Right? Same thing can be done with transgenic fish as well. Increase the yield, to increase the qualities, to increase the, or rather you say, improve the characteristics. Transgenic fishes are also being utilized. So in this unit of transgenic animals, we will be first learning about what are the methods to carry out the gene transfer. What are the different techniques that you can make use of, wherein you can make use of viral vectors, you can make use of micro-injection methods, you can make use of embryonic stem cell-based methods, some genetic modification systems, and then we will start taking examples of transgenic mice. Then in mice, we will see how models are generated, disease models are generated. Then what are the different things that you can do with these disease models. We will also see what is transgenic livestock, transgenic poultry. We will also see what are transgenic fish, right? Similarly, in the fourth unit, which is on reproductive biotechnology in animal breeding, again, something very interesting. And I think that this will also help you understand to a great extent what happens in assisted human reproduction as well. Because though the techniques might be looking way different from what happens in case of a test tube baby, what we today refer to as in vitro fertilization, but the principles of the techniques that we are making use of are quite closely similar. And it makes us understand also a lot of challenges and ethical concerns that are associated with this technology. We will come to that as we, at the end, as we describe the unit and reach the end of the discussion, we will come to the ethics aspect. So in this unit on reproductive biotechnology, we will be learning about the process of superovulation. There is something called a synchronization that you carry out. There is something related to embryo transfer technology as to how after in vitro fertilization is done, how do you transfer the embryos? Okay. What are the manipulation techniques for the embryos that you can carry out, which we call as micro manipulations. Okay. Then you will also learn about how sperm injections are done, which we call as intracytoplasmic sperm injections. How is artificial insemination done? Okay, how the semen is stored. Okay, because if you see, especially in the cattle, you will see this that they have this एस्पेक्ट सबसे एक्सपेंसिव अगर कुछ होता है ना ब्रीडिंग में कैटल में जो हमारे डेयरी इंडस्ट्रीज करते हैं जैसे कि अमूल हो या गोवर्धन हो गोवर्धन especially carries out the breeding of their own cattle. Okay, so they ensure that their breed has to be of the superior quality, so they don't let natural breeding take place. Okay, so they have this assisted reproduction that takes place there. So the most expensive thing to purchase in this kind of a setup is the sperm. So that's the reason why once the sperm is purchased, they have a proper technology to store those sperms because it may have to be used for multiple cycles. So that is purchased, kept in frozen condition, and that's used. These days in animal assisted reproduction, there's a lot of cloning aspects that have come into the picture. There is somatic cell nuclear transfer that is being carried out. And also people have started, if the commercialization is gender based, wherein the product is seen only in the female, then they have also started using technologies of chromosome separation to see that the offspring is of a specified gender because the product may be related to a specified gender. So it's gender specific. Now, the moment you hear this thing itself raises a lot of ethical queries, right? Can you tell me what could be the possible ethical concerns in both the reproductive biotechnology part as well as in the transgenic animals part? Can you raise some of the ethical concerns that come to your mind the moment you hear about this? Come on.
So the transgenic gene pool is mixed with the natural one, so it alters the natural genome.
Right. So one of the major concerns would be how is the newly inserted gene going to fare in the future? Is it going to be passed on? If it's going to be passed on, we are introducing a new gene which was not present in these set of animals at all, and we are letting it pass. That could be one of the biggest ethical concerns. So that's the reason why you will see that most of these animals, they are not let to breed. And once their work is done, they are normally euthanized or they are killed. Okay, because we don't want these new genes to enter the gene pool of these organisms because it's not a naturally occurring gene in that particular organism. Because we don't know what the consequence would be. It could be a human interference in the process of natural evolution. So we don't want to do that, right? Very good point, Harshali. Is there anything else that you guys can think of? Anything else that comes to your mind
It could cause
in
a
terms
lot
of
of
ethical
animal death.
concerns?
It could cause a lot of animal.
One at a time, please.
Payaswini, go ahead.
Priyanka, can you be louder?
Uh, yeah, actually, I was saying if Payaswini is talking, she can continue. Another, I think, ethical concern will be first if that's a male offspring, it will not be treated as such. Like I was at Govardhan once for IV, so I get to know the male spring. They either gave them to the villagers if somebody wants to keep, otherwise they don't let the offspring in the premises. And
Yes.
another is they don't keep them with their mothers. They literally separate them as soon as it gets birth. And
Very good.
another thing is the gender disparity that will arise, like the population of female will be a lot and the male will not be there anymore after some time if this continues.
Very good. Excellent. So we have to understand that if we are looking at gender specific reproduction, wherein we are making use of these chromosomal technologies, there's going to be a lot of gender bias that's going to be generated, because commercially only one sex is significant over here. So we are going to decrease the number of the others. Will that help us in any way? No, right? And specifically separating the young ones from their mothers so that the milk especially, but there are several dairy industries, because of animal ethics and ethical boards saying that the young ones have to be kept with their mothers, they have the right to have the colostrum, and then they need to be separated. Several dairy industries today follow that particular law, okay, because it's a heinous crime. Okay, but then we need to understand that it's all under in a country like us, wherein the population is so huge, how much control are we able to keep over all of these? Ethical concern are one of the biggest challenges, believe me. And that's the reason why, if any kind of research work is involving any animal model, there needs to be permission taken from the ethical committee. Please
Yes, sir.
understand that any animal which goes beyond the non-chordate section, I mean from the beginning of chordata, Urochordata, Cephalochordata, then you have the tetrapoda, which has your amphibians, reptiles, aves, mammals. For all of these, Pisces, for all of these, you have to have permits. For non-chordates, permit is not needed, which means if you're working with poriferans to hemichordates, you don't need permits. But if you're working from chordata onwards, you need permits. Now, the reason for this is that nervous system is prominent in chordata. And that's the reason why it becomes essential that you take permits, because these are the organisms which will be able to sense pain. That was a logic given. It in no way justifies killing the organisms of non-chordates, but this was the justification that at least for these, permit is a must. So every institution which has laboratories which works with animals, they need to have an institutional ethics board. The institutional ethics board will review the work, okay, the proposal, because until and unless you get your proposal granted, you are not allowed to start work with animals. Institutional ethics board will report to the upper authority, which could be a district level or a state level ethics board. That has to report to the national ethics board. So it's not that institution के लोग कुछ भी करेंगे तो उन पे चेक करने वाला कोई नहीं, ऐसा नहीं होता. There is a strict check. Okay, so you have to specify how many animals are you going to use, how many of them will you kill in the process, because in some experiments you have to kill the animals because you may be analyzing some organ inside them, so you may have to kill them. So you need to specify that in detail and justify, is it really essential? Because the ethics board will always try to convince you that is there an alternative way by which you can do the same study without killing or sacrificing an animal. Yes, Devendran, I'm sorry if I was interrupting you. I saw that you were trying to ask something, but I was in that flow. Devendran, you were saying something?
Yes, sir. No, sir. Actually, I just remember that chick culling was so high in recent times that it got a petition that it should be banned in many countries. And Germany and France were the first country to ban male chick culling, as uh, when the male chicks were born, after hatching, they were culled. They were killed as they don't have any use in poultry industry.
Exactly.
Yeah,
So, this is what we need to stop, because we as humans, we have started interfering with the natural process. Okay, if this happens to us, will it be agreeable? Right? So, this is what Animal Ethics Committee's first job is going to be, to understand, to realize that is it really essential for us to do this experiment? And if we are doing this experiment, What care are we going to take to ensure that we are not killing the animal if it is not needed to kill it or to sacrifice it? In several experiments, it may be just noticing what changes the animal shows, and it doesn't involve sacrificing the animal, right? So these aspects have to be kept in mind. And when we talk about animal assisted reproduction in animals, there are other issues or other concerns raised. Because if we start extrapolating these techniques on humans, okay, that's going to be unacceptable. That's going to be unacceptable because it will be used for all wrong purposes. I'm sure that somewhere in your curriculum of biotechnology, you would have learned that genetic engineering, genetic manipulation techniques, and with the advent of CRISPR-Cas, there is a huge fear that designer babies concept may be revived, which US was trying to practice to a great extent, but then their laws and regulations and human rights commission brought a hold on it then and there. But if we start extrapolating these techniques on humans, wherein a couple walks into the IVF center and demands that these are the characteristics that my baby needs to have, will it be acceptable? Imagine a couple walking into a IVF clinic, which is termed as designer baby, okay, and they telling that my baby needs to have long legs, my baby needs to have blue eyes. This is not acceptable because you are interfering with the natural process. And if it doesn't work properly, there is a chance that it may also go wrong, and the baby who would have naturally born would be born with some kind of abnormality. And you want that baby to be athlete, so you want him to have or her long legs, but the baby may not be interested in sports at all. So who are we to take decisions for these individuals? So same concerns and same questions are raised by people who are animal enthusiasts, that how are we taking these animals for granted? No doubt that these testings are very significant, but with these ethics guidelines coming into the picture, animal usage has been restricted to a great extent, and we are now trying to work the maximum with cell lines. And thank God to the bioinformatics area that a lot of wet lab work is now first tested out as computer-based simulations. You check if it is really going to work, and only then you proceed to animal model. So पहला एक जमाना हुआ करता था जब फार्मास्यूटिकल इंडस्ट्री में 1000 एजेंट्स जिनको पोटेंशियल थेरेप्यूटिक एजेंट समझा जाता था, सारे 1000 एजेंट्स को एनिमल्स पर टेस्ट किया जाता था, और वो भी तुमको डुप्लीकेट ट्रिप्लीकेट में चाहिए रिजल्ट्स, तभी तो तुम उसको स्टैटिस्टिकली रेलेवेंट कंसिडर कर सकते हो. तो तुम इमेजिन करो कि 1000 एजेंट्स अगर मैं ट्रिप्लीकेट्स में करने जाऊं और उसमें भी कितने तो पैरामीटर्स मुझे स्टडी करने होंगे, तो उतने सारे एनिमल्स को हम बलि चढ़ा दें. बट अगर मैं पहले इन एजेंट्स को कंप्यूटर बेस्ड साइमुलेशन से चेक करूं कि भाई इसका डॉकिंग कैसे होता है, कितने एफिनिटी के साथ ये मेरे टारगेट के साथ बाइंड कर सकता है, तो शायद 1000 का नंबर 10 पर आकर रुकेगा. So I'm still going to make use of animal models, but restricted to a great extent, right? So these kind of advent in technology is what has made things possible for us. But we have to always keep in mind that ethical challenges are going to be there, and we have to deal with them, because what comes first is life. Respect for life, and then all these technological elements. Because if we start behaving in this way, in a ruthless manner, we and robots are going to be no different. We have been given a brain, we have been loaded with emotions, we are thoughtful animals, as rightly said, so we have to use that because we are given compassion. So with technology's advent, ethical concerns have to be matched. I hope that is making sense to all of you. Okay, so these are the two units that we will be discussing. Now, I wanted to understand that I will be sharing with you guys a wiki link. Okay, I'll show you what a wiki is. I'm also going to post the link in the chat box. I will also share this on your email. Once I have all the email IDs with me, I can still see that only 11 people have filled it. I'm requesting others also to fill in the form. I will repost the form link. Okay, so I will show you till then what the wiki is. So let me cast my screen for that. Are you able to see the screen?
Yes, sir.
Great. So this is a wiki created for you guys, wherein there will be different folders like this. You have references, there will be PPTs, there will be recordings. So I'll be sharing all recorded sessions, whatever sessions we have, it's going to be recorded. I will share those recordings over here. Whatever presentations I use while teaching, I will share it over here. Under references, you can see already something is shared called as books. So when you click on books, it will take you to a Google Drive tab. Okay, so this is what will open up. I'm sure that you're able to see the Drive tab right now on your screen. Okay, so whatever books were prescribed in your syllabus for these 2 units, I have put all the PDFs over here. There was also a paper recommended in your syllabi, even that has been put up. There's only one book, BD Singh, जिसका e-copy नहीं है, but I personally do not recommend that book. It's a very bad book. But if there is something that I will be dealing with from that book, I will see to it that I'll scan those pages and I will upload the PDF over here. Rest all books I have put up here so that you can refer to them, you can read them, okay? And whenever we discuss a specific topic, I will also tell you that this topic is from which books. So you can note them down and read them for your preparation. I also make it a point that at the end of every lecture, I normally give a Google form link to the students, which has few questions. They could be MCQ based questions, one word, one line answer questions at times, wherein you will have to answer questions based on the concepts that we discussed in that lecture. They will not be used for your evaluation anywhere, but it is for you and me to understand where you guys stand and what you have assimilated from the session. So I hope you will take it positively, okay? And those assessment links will be given to you at the end of every lecture. And I will also make it a point to post those links on the wiki, okay? And those forms will be open for 24 hours, a day's time for you to fill. So for elective paper, as we are dealing with 2 units, we will be meeting on 2 days, Tuesday and Friday. While for the core paper, we will be dealing with, I think, on Saturday. Okay, so that's how we'll be meeting thrice in a week. And I would just suggest that, as we go ahead with our curriculum, please see to it that you are reading on a daily basis. If you have any kind of doubts or difficulties, please feel free to contact me. Okay, there is no issue. You can contact me on your Telegram, or you can also ping me on WhatsApp. This is the contact number that I have put in the chat box. Okay, in any way, if I can help you out, I'll be more than happy. So please feel free to ping me at any point of time if you have questions. I am also putting in my email ID in the chat box, so that if anyone wants to write to me, you can feel free to do so. Okay, so please do not hesitate. As I said, that every question is legitimate. Ask them, clarify your basics. That is what is crucial. I hope that today's session has given you an overview of what we are going to deal with. I am really very, very excited about the upcoming sessions, and I hope that we both get to learn a lot of things from these discussions. Okay, if you have any questions, please feel free to ask, or today being the first day, I think we can stop at this point. Any questions?
Sir.
No, sir.
Okay, great. So please fill in the Google form it would be great if you guys given your response so that I can send you the calendar invites for the lectures. I can see there are 19 people in the batch, but only 15 have filled it as yet. Others, please fill it immediately. And others who have filled it can now start disconnecting. Thank you so much.
Thank you, sir.
Really
Thank you, sir.
Thank you, sir.
Thank you.
thank you so much.
Thank you, sir.